Hello viewers, today we will start with elementary plant biochemistry and chemistry of plant products. Biochemistry is an important subject in plant sciences, and today we will discuss what is biochemistry, what is the scope of biochemistry and how it is linked with other sciences which are of plants. So, what is biochemistry? Biochemistry broadly deals with the chemistry of life and living processes. Biochemistry is a science which deals with the internal processes going on in a system. It may be a plant, it may be an animal. And as you know, the scientists knew that a number of chemical reactions are going on in the system, but the term biochemistry was first introduced by Carl Neuberg in 1903. So, the biochemistry is as vast as life itself. You know, in the plant system, every process, it may be germination, growth, development or production, every aspect of plant is composed of hundreds or thousands of the reactions inside the cell. And for these process, every step of plant is packed with hundreds of biochemical reactions. If we talk about the plants, everything is going on by internal processes like reactions, and these reactions need a favorable condition for water, for temperature, for relative humidity and for pH. And in the biochemistry subject, we learn how these ideal conditions are important to have a process finished in the system. So, biochemistry is the most rapidly developing and most innovative subject in medicine and plant sciences. This becomes evident from the fact that over the years a number of Nobel Prizes have been awarded for biochemistry. In the plant system, a number of processes like photosynthesis, respiration, transpiration, everything is governed by the biochemical reactions. In medical sciences also, the biochemist has played a pivotal role in developing the vaccines and cure of certain medicines. And most of the research in plants, animal sciences and medical sciences, they start with the biochemist, and that is why a number of Nobel Prizes have been awarded for biochemistry. So, in case of plant system, C3 cycle has been developed by the Kelvin, who was the biochemist, and he got Nobel Prize. C4 pathway, another important pathway in the plant system, was also developed by Hatch and Slay, who were biochemists and got Nobel Prize. Hans Krebs, who discovered the Krebs cycle, was also a biochemist who got Nobel Prize for discovery of this Krebs cycle. The biochemistry is an important discipline, and whenever we talk about the research in plant science and medical science, it cannot be completed without the biochemistry. The discipline of biochemistry serves as a torchlight to trace the intricate complexities of biology, besides unraveling the chemical mysteries of plant life. Most of the people know about the vitamins, carbohydrates, proteins, they all have been discovered by biochemistry. In plant system or in animal system, the importance of carbohydrates, their physical properties, their role in human diet, they all have been identified by the biochemists. So, the discipline of biochemistry is very important to unravel the chemical mysteries of plant life. You know, for example, proteins. We know that proteins are very important for plants as well as for humans, but what type of proteins are important? Whenever we talk about the plants, how particular proteins are accumulated in particular environment. They all have been identified by the biochemist. For example, the plants, they are so smart that they have the capacity to accumulate particular type of the adjustment in the plant system as per the environmental conditions or the resource conditions, and the biochemist with the help of physiologist and biotechnologist can understand that what type of the accumulation has been done by the plants. So, when we talk about the research in plant sciences, we think to develop the plants which have the capacity to biochemically adjust their chemical reactions. The biochemical research has amply demonstrated that all living things are closely related at the molecular level. Actually, we say that the biochemistry starts when the physiology ends. Similarly, the biotechnology starts when the biochemistry ends. So, this biotechnology provides a way to work on the molecular tools or molecular level. The biochemistry is the subject of unity in the diversified living kingdom. The biochemistry provides a path to molecular biologists that what type of research they should do, what techniques they should employ for the betterment of the life for the society. So, the biochemistry has reached in advanced phase now. The biochemist has provided the tremendous impact on human welfare and have largely benefited mankind and their living styles. Insulin is one of the example that how the discovery of insulin by biochemist has affected the society in a big way. The application of biochemical tools for identification of plant diseases as well as quality parameters in medicinal and aromatic plants is the another way to show the importance of biochemistry in the society. A number of vaccines has been developed by the biochemist to cure the diseases like polio, and nowadays they are working to cure the cancer also by vaccination. So, in case of the plants, nowadays we have vaccinated banana, we have some other vaccines through plant system which can cure the diseases. Okay. The biochemistry has a link with other sciences. Whenever we talk about the plants, the plant physiology, seed physiology, biotechnology, genetics, genetic engineering, they all are the basic sciences which have the linked to each other. In this consequence, biochemistry is an important step to get products through biotechnology and genetic engineering. Some of them include growth hormones, interferon and transgenic plants. In the due course of this module, we will learn that how different hormones they affect the plant growth and development, how vitamins affect the plants as well as human bodies, and how the scientist has developed the transgenic plants by employing the techniques developed by physiologists, biochemists and biotechnologists. So, for developing such transgenic plants or for developing the growth hormones, the biochemist has played a great role with physiologists and biotechnologists. You have learned about the flavor saver tomato, where the reactions of ethylene has been stopped by the physiologist by understanding the biochemical reactions of ethylene biosynthesis. Similarly, a number of transgenic plants like Bio902 has been developed in Brassica with the help of the biochemists. So, the biochemistry provides a way for molecular biology genetic engineering. Without biochemistry, without knowing the biochemical reactions inside the cell, it is not possible to generate the transgenic plants. It is not possible to do plant biotechnology without proper knowledge of plant biochemistry. Plant biotechnology is the techniques which has been developed for the welfare of the humans, and this technology starts with the physiology and biochemistry. And biochemists have identified most of the reactions inside the plant which decide the fate of a plant, and biotechnologists utilize these techniques, these metabolism to convert or to utilize for betterment of the society. So, the biochemistry is a fundamental subject, and the story of biotechnology and other sciences cannot be completed without the use of biochemistry. The biochemistry has links with other sciences as well. For example, medical sciences. In medical sciences, the biochemistry has played a pivotal role in developing a number of drugs and antibodies and proteins to cure various diseases. For example, the proteins has been developed to cure the blood sugars, cancer and anemia, and nowadays some of these proteins are being utilized to cure these diseases. One of the best example about the use of biochemistry in medical sciences is the insulin. You know, insulin is very common to cure the diabetes diseases in humans, and this insulin has been developed by recombinant DNA technology by biotechnologists with the help of the biochemist. One of the recent example of the biochemistry in medical sciences is the use of hepatitis B vaccine. You should know that India is among the 4 countries who has indigenously developed the vaccines of the hepatitis B, and among these countries USA is the top and India ranks 4. And from 1997 we are fortunate that our country has started preparing the indigenous vaccines for hepatitis B. In case of veterinary sciences, you must have heard about the dolly sheep. This dolly sheep was developed by utilizing the biochemical and biotechnological tools, and this dolly sheep does not have father, it has only mother, and its mammalian cells have been used to develop this dolly sheep by using totipotency, that is a tissue culture technique. In case of agriculture, you know, there are number of proteins and there are quality parameters which we want to incorporate in the plants in our food products, and the biochemistry provides a way to identify the qualitative parameters or qualitative characters of in a particular crop. And like in case of wheat, we know that different types of proteins are available, and which type of proteins we need in our crop, that can be decided by utilizing the biochemical tools. So, it shows that the biochemistry has a close link with other disciplines in agriculture, medicine and veterinary sciences, and without biochemistry we can not do the advanced research in these areas. Now, the question is how biochemists do all these activities, what are the tools, what are the techniques which are utilized for this type of the research. As I told, biochemistry is an experimental rather than a theoretical science. Understanding and development of concepts in biochemistry are a result of continuous experimentation and evidence obtained therein. So, whatever we are telling, whatever we think, that is dependent only with the results and experimentation. It can be stated that the foundation for the present and of course for future knowledge of biochemistry is based on the laboratory tools employed for biochemical experimentation. And nowadays, we have good labs for biochemistry, good instrumentation. With the help of them, we have the repeatable result of the experiments. So, among the biochemical tools, chromatography is one of the important tool. It is an analytical technique. It is an analytical technique dealing with the separation of closely related compounds from a mixture. These include proteins, peptides, amino acids, lipids, carbohydrates and vitamins. Actually, what happens? We know that in a plant system or animal system, proteins are available, peptides are available, amino acids are available. Biochemistry tools provides us the information that which type of proteins are available, how much quantity is available, what is the ratio of different types of proteins or amino acids. And this chromatography is an important tool which separates these biomolecules in small portions. So, chromatography usually consists of a mobile phase and a stationary phase. The interaction between the mobile and stationary phase results in the separation of the compounds from the mixture. The chromatography are of different types like partition chromatography, adsorption chromatography, ion exchange chromatography, affinity chromatography, TLC and HPLC. These TLC, that is thin layer chromatography, and HPLC, high performance liquid chromatography, are widely used in the biochemical laboratories. The electrophoresis is another tool widely used in the biochemistry. The movement of charged particles, that is ions, in an electrical field resulting in their migration towards the oppositely charged electrodes is known as electrophoresis. Electrophoresis is a widely used analytical technique for the separation of biological molecules such as proteins, as well as to visualize the bending pattern of DNA. This technique is dependent on the principle of mass and charge ratio. Nowadays, a number of biotechnologies, they are using this technique for separation of the bands in different species in the crops. Now, come to another very primitive but important tools, that is colorimeter and spectrophotometer. Colorimeter is the instrument used for the measurement of colored substances. This instrument is operative in the visible range, that is 400 to 800 nanometer of the electromagnetic spectrum of light. The spectrophotometer primarily differs from colorimeter by covering the ultraviolet region, that is 200 to 400 nanometer of the electromagnetic spectrum. It is more sophisticated than colorimeter. Both these colorimeter and spectrophotometer works on the principle of Beer-Lambert's law. These tools are dependent upon the color intensity of the product. So, you know, a number of molecules like proteins, amino acids, sugars, they lie in different ranges, that is 200 to 800 nanometer, and depending upon the nature of these molecules, these colorimeter or spectrophotometer can be utilized for measurement of the intensity of a particular molecule in a particular region. Flame photometry. The flame photometry primarily deals with the quantitative measurement of electrolytes such as sodium, potassium and lithium. The instrument used for these measurements is known as flame photometer. This flame photometry has wide utilization in plant sciences. You know that there are 16 elements being utilized for growth and development of the plants, and these plants need these elements in a particular concentration at particular stage. So, with the use of this flame photometry, we can know that which element is present in the plant and what is the concentration and what is the stage. And basically in the flame photometry we are able to estimate sodium, potassium and lithium. Nowadays the advanced version of flame photometry, that is AAS, atomic absorption spectrophotometer, is available, where we can measure a number of nutrients, essential as well as beneficial elements in the plant system. Now, radioimmunoassay, we call it RIA, RIA. This technique is used for the estimation of compounds in biological fluids that are found in exceedingly low concentration, that is in nanogram or picogram. RIA is highly sensitive and specific analytical tool. This technique has tremendous application in the diagnosis of hormonal disorders, cancer and therapeutic monitoring of drugs. This RIA is based on the radioisotopes of the elements. Now, ELISA, enzyme linked immunosorbent assay, this is the advanced version of RIA. In this, it is a non-isotopic immunoassay. An enzyme is used as a label in ELISA in place of isotope employed in RIA. ELISA is as sensitive as or even more sensitive than RIA. Further, there is no risk of radiation hazards in ELISA. So, whenever facilities are available, we prefer to do ELISA rather than RIA. This ELISA technique is widely used for the determination of small quantities of proteins like hormones, antigens, antibodies and their biological substances. The most commonly used pregnancy test for the detection of human chronic gonadotropin, that is HCG, in urine is based on ELISA. ELISA can also be used for the diagnosis of AIDS. So, you can understand that how these tools are applicable in the diagnosis of human diseases like pregnancy test and AIDS. Dear listeners, so in the course of this modules, today we have discussed about the importance and scope of biochemistry. We have learned how biotechnology helps to understand the internal process of the plants, animals and veterinary sciences. We have learned that how biotechnology has helped in the development of other disciplines. We could know that how biotechnology cannot be done without the help of biochemistry. The generation of many diseases and their cure has been, the biochemistry has provided a way to cure many diseases in the animals as well as in the plants, and a number of drugs has been developed with the help of biochemistry. I hope that you have enjoyed this lecture. Thank you.